We live in a diverse world, rich in abundant plant and animal life. Differences between populations can give us information about the health of an environment, or just alert us to the different environmental conditions.
Measuring the abundance of populations in situ can help us understand the changes in populations. For example, the data can help us to determine if there's any abiotic factors that impact on the population.
We're students here from the Sixth Form College in Colchester, and we're here at Highwoods Country Park. This site is a south-facing sloping field with a diverse range of plant species. There's a pond just to the bottom right of this field. Walking around, we can see evidence of buttercups, ribwort plantain, birdfoot trefoil, and a range of grasses.
But do populations of these plants differ from the top of the hill to the bottom of the hill? We're here to investigate whether there's a significant difference between the number of buttercups at the top of the hill compared to the bottom of the hill. We'll also investigate whether there are any differences in the abiotic factors that might help us to explain why there might be a difference in the number of buttercups in the two locations.
We can see two species of buttercup in this field. There's the creeping buttercup, which can be identified by its spreading runners and its leaves divided into 3 lobes. And there's the meadow buttercup, which does not possess any runners, but has more sharply defined leaves divided into 3 to 7 lobes.
Buttercups tend to prefer damper sites. Looking at this site, we expect that damper conditions are likely to be found at the bottom of the hill, so we'd expect to find more buttercups at the bottom than the top. But how can we find out if this is correct? What factors might affect buttercup populations at the top and the bottom of the hill?
Factors which might impact upon the populations of these plants include light intensity, humidity, wind velocity and air temperature. These can all be measured using a multimeter.
We also want to know whether there's a link between abundance and soil factors such as humus and water content. We can do this using a soil auger.
We can't count every single buttercup, so we're going to have to carry out random sampling methods, here at the top of the hill and down there at the bottom of the hill. We've marked out a 10 metre by 10 metre plot, and by using a random number generator, we will identify where to place our quadrats.
Next, we record the number of buttercups in each quadrat, take soil samples and take measurements using the multimeter. Whilst we collect the data, we calculate a running mean of buttercups to make sure the data we're collecting is representative of the population. When adding data has a very small impact on the running mean, we can be sure we've collected representative samples.
Having done our sampling at the top of the hill, we now need to do it again at the bottom of the hill.
Now that we have our data and we've collected the number of buttercups at each site, the wind velocity, the humidity, the air temperature and the light intensity, and we've collected our soil samples, we can now analyse the data. We're looking to see if there's a significant difference in the number of buttercups that will support our hypothesis. We hope there will also be differences in the abiotic factors to explain our findings.
Now over to you. Download our data from the Big Picture website and try it for yourself. If you need help with deciding what statistical methods to use, go to the number crunching issue of our website. All resources are available from bigpictureeducation.com.